everybody. I'm Lisa Miner. I work with Multispan. Multispan is a company in California whose main objective is to build cell assays, develop cell lines, and the cell lines and assays are for the large part for GPCR targets. Today I have here to share this talk with me, Terry Kanakin. So Terry, can you introduce yourself?
Hi, folks. Yeah, hi. I'm Terry Kanakin. I'm a pharmacologist who's interested in receptors, how they work. I worked in industry for 32 years, 25 at GSK, but I'm currently at UNC School of Medicine. But I'm a great proponent of the power of assays to um, demonstrate drug effects.
So thank you, Terry. So today, what we're going to do is discuss optimal assays for driving GPCR, ligand, and antagonist drug effects. So most of us realize or know that are in the field that GPCRs are a really well-worked target for drug discovery. A large number of small molecule drugs are GPCRs, are targeted to GPCRs. But since the early discoveries of GPCR targets, a lot of things have become known about GPCR signaling and how complicated it is. And so today, we're going to discuss, in light of all of this information, what are the best assays for looking at GPCR activity and how we decide which assays are the best to suit our needs. So again, what are the most effective assays to test unknown molecules for activity against GPCRs? And the reason for this is because all of the different assays that have become known recently, all the information, new information and relatively new information regarding GPCR signaling. And essentially, we want to make sure that as you're building an assay and as you move towards screening for compounds directed against GPCRs, that you get data that is the most meaningful and that suits your needs. So that being said, Terry, explain to me what are the best assays for measuring activities at GPCRs?
Well, if we think about the history of all this, historically, programs would choose a binding assay to get affinity, and then a highly sensitive functional assay to detect efficacy. And these worked. You would get answers like my compound binds to the receptor, and my compound does or does not produce response. But they are actually quite limited, and there are now better assays to answer those questions and much more.
So Terry, can you describe the binding assays and what's good about binding assays? What's it give you versus what a cellular assay gives you?
Well, binding assays, of course, show you a physical interaction of your compound with the target, which is good. If you're interested in a quantitative number that reflects what you'll see in physiology and pharmacology, they can be limited because binding assays are dependent on a number of experimental conditions that can change the numbers that come out of them. For example, slide 1 shows you the relative effects of receptor signaling protein stoichiometry. Unless you're using the exact assay that reflects your functional response, then you have a different membrane receptor to, in this case, G protein stoichiometry, and that can affect your binding assay. As you see in the slide, we have a binding displacement curve with amylin in a receptor overexpressed system. And what you see is a complex curve. It's what happens is you begin to see the high affinity state, and then as the receptor runs out of G protein, you get the low affinity state. And so you get curves that are very difficult to interpret. And certainly the numbers that come out of these kinds of assays can't be applied to a functional assay. But there are other things that can cause binding assays and true function to be different. For example, next slide shows if you have an allosteric compound. So now we have a compound like this is an ester. It's a non-competitive inhibitor of glutamate receptors. And what you see is in functional assays, it inhibits response. In a binding assay, it does nothing because it's binding to a separate site on the receptor that doesn't interfere with the radioligand binding site. And so these kinds of compounds you can't study with binding. And the next slide shows a general issue with this. The protein species that a binding assay monitors are different from the protein species that govern functional response. And of course, response is what matters. So a binding assay will tell you what protein species are involved in binding of the radioligand, but a functional assay tells you about what species control response. So in general, binding assays can be useful in a general way that your compound interacts with the receptor, but they are highly limited and capricious with respect to the affinities and the numbers that come out of them.
I guess part of the issue could be too is how we have to jury rig the binding assay so that you have enough receptors on the surface to actually be able to measure an effect. So you screw around with your system to make it super non-physiological, and then you expect it to give you an answer that's meaningful in physiology. Yeah. It's kind of a tough thing. So yeah. So I understand, and I think binding assays are really good standard assays. Downside is they're radio labeled, so you still have to deal with isotope most of the time, which is an issue. But I think that basically there are better assays that we can use versus binding assays. So a question for you is, what can you suggest? How can cells control sensitivity to hormones and neurotransmitters, and can that be used to build assays for signaling?
In short, yes, it can. If you had or have a functional assay that has low sensitivity, and then you have another such assay that has high sensitivity, you can pretty well capture just about everything that a ligand can do. So one way to modify receptor sensitivity or cell sensitivity is to alter the level of expression of receptors. And so the next slide shows what happens if you have a system with a low receptor expression level. You will get a partial maximal response, you will get a curve, and then as you increase receptor expression, you will go up to the window of the assay, and then the curves will start to shift to the left. It's those initial curves of low sensitivity that can be extremely useful. And I illustrate that in the next slide. So,
Slide 5. Yes. Okay.
yeah, slide 5, thanks. And so, if you look on the right panel, you see a low sensitivity assay and the curve to an agonist. And the maximal response is not the complete window of the assay. This is the ideal assay because it has the maximal response is a measure of efficacy, and the EC50, the midpoint of the curve, is a good measure of affinity. So you have two measures that come out of that kind of assays, affinity and efficacy. Contrast that with, if you look on the right, you see the same agonists in a system of high sensitivity. So you get nice curves, and they're all of equal maximal response. That's because you exceed the window of the assay. But the only measure of relative activity you have is potency. And potency, the EC50, is a complex amalgam of both affinity and efficacy in this kind of assay. So the only thing you could tell a chemist is, here is the relative potency of your compounds. And whereas if you were to run the experiment in the low sensitivity assay, you could tell the chemist, here are the relative affinities of your compound, and here are the relative efficacies of your compound. And that's telling them a whole lot more. And I can show you what that means in the example shown in slide 6. So here we have 4 bronchodilators, and I've made them so that they're pretty well equiactive. Their EC50s are all pretty well equal. There is no structure activity relationship for the chemist. However, if you were able to dissect the EC50s into affinities and efficacies, you would see a completely different picture. So let's run those compounds again in a low sensitivity assay. Slide 7 shows, indeed, there is a rich structure activity relationship. What's happening is affinity changes with structure and efficacy changes with structure. So you've converted an assay that's giving you nothing that'll help a chemist into something that is very effectively showing changes in structure having effects on affinity and efficacy. Now, I talk about affinity and efficacy as if they are separate properties of drugs, which they are. Slide 8 shows us a famous example where this was illustrated. Jim Black was trying to make compounds that inhibited H2 receptors for healing of ulcers. He wanted to block histamine. And he realized that the SAR, structure activity relationships for efficacy and affinity, are different. So we had the SKF chemists start with the autocoid, histamine, which he knew interacted with the receptor. He had separate assays for affinity and efficacy. And as SKF made analogs, made compounds that reduced efficacy and increased affinity. And that's how they got to the H2 inhibitors. So that's a very famous example, resulted in a Nobel Prize and a very effective drug. And it just illustrates how much better it is if you can give chemists such textured um, data on the activity of their compounds. So I've centered on low sensitivity assays, hopefully illustrating their use. What about the high sensitivity assay? That's useful as well because it's going to pick up your efficacy of very weak compounds. Slide 9 shows you an example. This is an allosteric potentiator, BQCA, and in a low sensitivity assay, it potentiates acetylcholine. However, when you test this in a high sensitivity assay, it basically unveils another activity, and that is direct agonism of this compound, which in this case could be extremely useful because it could lead to side effects. So having these two kinds of assays in dual use could be extremely useful. You'll get affinity efficacy and you'll pick up low values of efficacy.
So if you were screening, which would you do first? Would you do a low sensitivity system where you're going to pick up absolutely everything? Actually, yeah. No, I get them mixed up, high and low sensitivity. Would you do one that has low receptor expression first, or would you do one with high receptor expression, so that you're picking up everything in the world?
Yes. Actually, that's sort of the standard approach. You start your screening assay should be a high sensitivity assay. It's just there to detect interaction. You don't know quite what it is, what the affinity is, and so forth. But you pick out the compounds that choose the receptor. Then to characterize it, you would go to a low sensitivity assay. Yes.
So that you would be able to tune out or tune in what is
Yeah.
the nuances of your receptor and of the ligands.
Yep.
Let's, so how do you know which is the right setup for you when you are in vivo? I mean, that's one thing we've asked a lot is, do we ever know what our actual tissue needs and what kind of effect we need to have in the tissue, or are we just still in that exploratory phase?
That's to anyone who's worked in industry, that's a well-known question. What
Yeah.
will this compound do in vivo? And truly, the honest answer is I don't know. But what you will be able to do is prepare yourself with knowing the limitations of your compound, because as you put it in vivo, it's going to encounter a wide range of tissue sensitivities. And the worst thing you could do is constrain yourself with one snapshot of this is
Yeah.
what this compound does. And then you're surprised when it meets a tissue where it does something else. So I think this low high combo of assays should pretty well equip you for just about anything you might expect in vivo.
Well, that makes a lot of sense. And actually, testing molecules in vivo that have different profiles would probably be very helpful and beneficial to understanding how it actually affects your physiology.
Yep.
So let me go into what multispan can do for clients and what we've done for clients with respect to receptor expression. So we provide cell lines for people. We either make them for for new targets, or we have ones that we have in our for sale that are already part of our catalog for targets now. And the way that we actually develop these are we look at functional assessment of activity, and we always look at functional assessment activity to pick our clones. So in slide 10, what we can see is why this is really important. The bottom curves are looking at FACS analysis. All of our receptors are tagged with an N-terminus tag so that we can monitor the surface expression of those receptors. And we can see when you look at those curves, the dark line is the tagged receptor, the light line is the parental cell line. So what we can see as you increase receptor expression by going from left to right on this graph is that as you increase receptor expression, you wind up losing or changing the ability of the functional signaling of that cell. The top is the functional signaling, and we have 3 different ligands: dopamine, phenoldiprime, and bromocriptine. And what you can see basically is that the one ligand, the bromocriptine, is supposed to be a partial agonist. When you have almost no receptor expression, you don't even see it. It doesn't show up at all. When you have partial receptor expression, you actually see now it's a partial agonist. When you look at increased receptor expression, now it becomes a full agonist. So this is one of the reasons it's really important to measure this type of activity. And the cell lines that we provide, if we're making one for you, we have both high and low receptor expression. We maintain both, and you can see which one works best for your use. And we also have ones in the catalog that are high and low expression. So that's if you're interested, you can look at our multi-span catalog or give us a call.
That's extremely important. Assays, the team that has assays that can see things no one else can see has got a distinctive advantage. And so being able to tune assays lower and higher activity is a very powerful tool.
Yeah, we found that, and we found that our clients think the same thing. The people keep coming back to us for all sorts of different things. So I think that's a good indication that they at least like what we do. So let's go on to the final slide of this presentation, which is benefits of high and low sensitivity functional assays. So
right, yeah. So we've it's quite simple. Low sensitivity assays enable you to directly measure affinity, which is something you would get from binding, but it would be a more physiologically relevant binding number in terms of an EC50. You also get a good measure of relative efficacy from the maximal response of the curve. So you can give chemists a textured structure activity relationship for both affinity and efficacy, which can be different and which can be more detailed and informative than just something like an EC50. Then again, if you have a low, very high sensitivity assay, you can cover the other end of the scale. Compounds that are, say it's an antagonist that doesn't seem to have efficacy, when you put it in this kind of assay, you may reveal that it does have efficacy. So you will detect low levels of activation that could be relevant in vivo. So in having these 2 kinds of assays pretty well covers your whole spectrum.
So thank you very much. Is there anything else we want to talk about on this episode?
Well, yes, a important area that has emerged with GPCRs that has basically revitalized the field is the area of signaling bias. Previous to understanding this, it was thought that efficacy was a mono-dimensional signal varying only in volume. Now we know it's a complex mixture of various pleiotropic signals, GI, GS, G protein, beta arrestin, and so forth. And receptors are not switches that just turn the volume on and off. They can discern what pathways they would like to signal to, and this is a function of allosteric receptor structure. And so different compounds make different receptor conformations, and those translate to different signaling patterns. So bias is really the emphasis of a signaling pathway at the expense of others. So now you can have a compound, two compounds interacting with the same receptor, but one may signal differently than the other. This is a tremendously powerful tool therapeutically because we can, in theory, tune out bad signals, emphasize good signals, and improve therapy.
That's really interesting. That actually is going to be our topic for our next presentation. So for anybody who wants to get more information, please contact either Terry or me, or multispaninc.com for our contact information, or for what Multispan can do for you. And thank you very much, Terry, and I look forward to
Thank you.
talking to you in the future.
Thank you.